we said if you start with the matrix that we wrote really last week, this is transition matrix. So what is the rate at which you go from here to here? You have 0 molecules that are bound, and you have total m molecules. So what is the rate of having one molecule bound? You need to take the rate of binding, which is lambda, the number of total number of possibilities m, and the total number of free binding sites is s. There is nothing bound, right? And so you can write the rates like this. So this was one.
M is fixed here, or is M decreasing?
Yeah. 1 sec. And so the second direction of the matrix is that when m decreases, and so now we enter into the part of the chain where now you can have 0 bound, and one particle disappeared, and this happened with a rate which is exactly given by the Poissonian rate, which is mu multiplied by the total number of available molecule, which is m. All right, and we can continue now. Suppose in this line, no molecule disappeared, and we just get binding. And we arrive to here with T and M -1, correct? And again, once you're here, you can calculate the rate of transition from here to any of these. And up to where are we going? We need to go up to so if we have m molecules, so this is a state that interests us. We have T-bound molecule, T-bound molecules. Now, when is this going to fail? If the number of bound molecules, I mean, if there is not enough molecule to reach the threshold T, then wherever we continue, we'll never reach, right? So let me write down the matrix. So we have then 0, and then we end up here in 0 and T. And so we can have here, so the number of molecules has decreased up to the threshold. So which means that below this threshold, I will never have enough molecules to bind. So then this goes to, now we have binding. So we have 1 and T, and so on. And then we arrive to here, to the total number of bound molecule, which is going to be T, when we are left with exactly T. Right? And so what is the ensemble of state we're interested in? Is going to be these ensembles. So we start, this is a starting point. We start here. And now we let evolve the process. So now how do you end up here, here, or here? So how you end up here, this is not very hard. You just start here, you have no jumps. Just continue this way. How do you end up here? This is more complicated because what can happen? If you want to end up in here, there are many ways. You have to count all the paths that can go from here to here. So remember, that there is no way when you're bound to come back, and when you disappeared, you cannot appear again. You are dead forever. So for example, how you end up in here? You end up in here by any choice. You go from here to a certain point, Then you make a jump here, and then you continue this way. So how many possibilities there are? As many possibilities as there are to come once from here to here, which is exactly t plus 1, right? And so now when you are 1 and 2, then you have to count all the pairs at which you can jump in here for the first time, then you wait and you jump for the second time. So this you can write down. So I don't know if we wrote down last week the entire Markov chain.
If we're assuming we're in that final state, Tm or Tm minus 1, are we still evolving the system so we can go from Tm to Tm minus 1 down?
You can continue here, but
I was thinking, because obviously if you start off right at the top, you end up at Tm, are we saying, right, we're stopping there, and we're going to count everything we've got now? Or we're allowing it to evolve?
you're allowed to continue, but what I'm saying is that for the calculation, I just need to calculate the probability of those states. And actually, I am interested in the sum. So what we're interested in, what's the probability? So to answer your question, let's understand what are we looking for. What is the probability of ever reaching the threshold, that is the number of MS molecules when you finish your process? It's all the molecules are not gone. That let's call MS infinity, so those are the molecules that are ultimately bound, equals a threshold T. So this is exactly the probability, this is the sum of the state here, this is the sum of the probability of ending up with T molecule and having exactly M no loss, plus, so let's write it the sum, plus the next one, you have T, and there was M minus 1 up to infinity, and then we go up to PT and T. This is what we are interested in calculating. So obviously you can continue, but you know there's not going to help to calculate this, because once you are in here, it doesn't contribute. All right, so for example, again, I forgot if I'm, okay, I can write just the middle term for the transition probability, which is P, K, and M. I think we wrote it last week, but let me write it in any case. So last week we looked at if you started at something in the middle here, what was the transition from the guys above on the left, and where things were going, it's the transition from the right or below. So you have to write the transition probability for a two-dimensional Markov chain. And so now we use the fact that you have, in all cases, every transition is Poissonian. So you can, if you start from K minus 1 and M, this means that you have to have one binding. So you start with S0 minus K plus 1, M minus K plus 1, right? So this is the same as in here. What we said that to bind, really it's proportional to the number of free sites multiplied by the number of free binding sites, which is here, for when you start from K minus 1. Then there is the case where you are going to, particle is going to die, M plus 1 minus K, and you start from M plus 1 molecules, and there was no binding, minus the probability that nothing happened, which is S0 minus K, plus mu, multiplied by M minus K, P K of M. And this is true, so now we have to be very careful. This is true for T minus 1 between M and M0, and K between 0 and T. And so last week we noticed, I did it very carefully, I don't want to spend a lot of time on this, we did very carefully what happened when we have, what are the boundary conditions when you reach, when you're imposing a threshold. You remember last week we said when we have ms molecule equals to T, you can start with one bound molecule, then you go to make a transition to 2 and transition to T. T is a threshold. So in principle, as you mentioned, it can continue up to m molecules, or total m, or the minimum of m and s. So all the sites here are filled. Let's say s, let's suppose s is less than m. But if you impose a threshold, which means that your system stops when you reach the threshold, you have to modify the Markov chain such that this rate that is coming back is not 0. Because when you are in this state, you become absorbing. It becomes an absorbing state. So this k minus 1 becomes 0. And this is how last time we derived the first time to a threshold by putting this rate to 0. So here's the same. Now we have to calculate this, so you have to give other conditions, which I'm not going to write, just this is the Markov chain. Now you can look at the solution of this Markov chain going into infinity, where you know that in the middle, everything will be 0. If you are in this state, this is an absorbing state, so this p infinity here is not 0, right? So either you are here, or you end up in the rest here, which means you didn't reach the threshold. And so you can, again, I'm not writing all this term for the boundaries. But what is important is the normalization, that the sum of pk and m has to be equal to 1 for k equals 0 to n0 and m equals 0 to n0. All right. Okay, so now let me tell you how do you calculate this quantity in a more just with the hand-waving argument, and the calculation can be looked into the paper. So how do you calculate the probability of the first one, for example? What's the probability of this? The probability of this is once you start here, that you made a transition from here to here. So this is the probability. So this is going to be the ratio of the rate here divided by the sum of the two rates, which is the probability of making a transition from here to here. And then you repeat this up to here. So the probability of ending into this so I would like to erase this Markov chain. And you will see, I mean, you can, again, solve all the Markov chains, which is systematically can be done using elementary calculation. But let me give you, so the argument, the intuition is correct. So the pt infinity is simply the product of the binding rate. So lambda s minus I. You make the product from I 0 to t minus 1. And then divided by mu, so this is the rate here, mu. Okay, there is an m that I have cancelled everywhere, msi, sorry, mu plus lambda s minus I plus mu. Right, in principle the rate is, there is an m here in front of everybody. So that's the rate of transition from the first one is lambda ms, but then I moved the m out. Right? So it becomes this. So this is the rate, this is the probability each time. What's the probability? You take the ratio of this rate divided by the sum, which is the probability of making this compared to this one, and you repeat it, you multiply this by this term. Okay, and now how do you get the other ones? The other ones are basically can be derived as this product here, except that somewhere you're making a transition this way. So which means that you have to choose the index at which you switch from the first line to the second one. So this can happen at any of those lines. So you have to sum over all these possibilities. So this will give you the second term. And you do the same for the last one. So ultimately, this probability can be written in terms of this P infinity. This is PTM. PTM. Yeah, PTM 0, okay, whatever M, infinity, 1 plus, and then you get the sums over all possibilities, which is the sum over I1 between 0 and t minus 1. And then you have the transition which come from the mu term. So again, there is an m that will cancel. So this is mu divided by lambda s minus I plus mu. But this is for the first one. And then for the second one, you're going to get here a double sum, which is A1, 0. So of course, once you make a jump here, you cannot go back. So the second index has to be above the first one. So A1 less than A2, and t minus 1, and then you get this mu, the product for ik of the mu lambda s minus ik plus mu. And until you reach to the last one. All right, and so you end up with something like this, which is very unpractical to calculate, except in some limit where s is much bigger than the threshold, so the I here really do not count. And then, if you don't care about the I, it's really like throwing m particle and looking at the distribution of having t out of m particles that are bound. And then this is simply your binomial distribution. So this is what you can find, is that in some limit where, so I think it was t divided by s, which is large, s is much bigger than the threshold, then this total, let's call it total p. P is this quantity. P here is simply just the sum of the binomial distribution L and K. When you start, obviously, for at least t molecular bound, you need to have at least t here. And you can have, of course, t plus 1, so whatever. So this is t and m. And then this is the rate, the ratio of this. So the probability is lambda s plus mu. This is binding versus killing to power k. And then this is 1 minus this. So this should be mu divided by lambda s plus, yeah, to power n or m minus k, which is really the Bernoulli distribution. All right, so this is what I wanted to say last time, that this isn't practical to calculate, but at least you can put that in the computer. Last time, what was very nice is we could give explicit expression for this time. You can also calculate another interesting thing, but I don't want to do that now, is now we calculated the probability for this to happen. Now you can calculate the conditional time for this to happen. That is, conditions that you have reached those states, how long it takes. And then, so you have to look again at all the paths. So once you have the probability for one path, you just have to, it's not very hard, you just have to use the time. So the time for this is 1 over the rate. So you just, each time you have a probability, you multiply this probability by the time, and you end up with the mean conditional passage time. All right, so I think I'll stop here for this. Again, this was from last week class. We have many examples where this was relevant. One was for cell division, where we looked at the kinetochore, which is a process when cells have to divide. You have to make sure that the chromosomes are well aligned, and you have a finite number of them. I don't know, depend on the species. Let's say you have 16 or whatever. So you have a finite number, and you want to make sure that they are all attached. I mean, the cell makes sure this, and then they can start dividing. And again, this is a mean time to threshold, because you want to make sure that this threshold is reached before you divide. And we use this approach to estimate rate constant from data. So this was one application. I mentioned the application about synapses at the beginning. And the third application that we had on this process was the work we did with Kevin Barrage, about the probability that a messenger RNA would escape the nucleus protected by microRNAs. So again, you had DNA, a messenger RNA. The messenger RNA had some binding sites. So you ask the following question: how long it takes for this to escape through nuclear pore, small hole, here, before it is degraded by a killer, whatever the killer molecule is. So if you have, let's say, all of these molecules bound here, it is destroyed. So no way, no chance to go out. So there was in the game, a molecule that was actually protecting. That is, if the blue molecule is here, the red cannot come, and so it is protected. And so the idea, again, if you have a threshold, for example, if you assume that when the number of red reach a threshold C, that is threshold T, it is destroyed. So now you can have competition of this blue molecule that can come by diffusion and can be removed by detachment. At the same time, you have the red one that can come and leave. So the blue and red compete for the sites. If the red reaches a threshold, the game is over. What's the probability that you reach here before your threshold is reached? This is exactly the type of framework we've developed here. I don't have time to, it will be an additional hour. So what you have to look at here is the joint probability of having the total number of sites and the number of red crosses bound. Assuming that you have enough of them so that when they are released, they are in excess. So we ended up calculating this probability, the time it takes to happen, and it turned out that we could do some analysis, for example, when there was no unbinding. When you have unbinding, it's very hard. We didn't know how to calculate explicitly how it depended on the number of sites, and we had to use simulations. All right, so let's stop here for this mean time to threshold. There is still open problems in terms of the asymptotics. Here it's unpractical. Once you go to this pass and counting pass, it's very hard to get asymptotic formula in closed form, or where you can see the dependency in the parameters. Yet, for the case of plasticity, we are still very much interested in how to calculate the time that a certain number of molecules have reached a threshold, especially when molecules are not completely independent but are organized in a ring. That is, you can have molecules like this. This is the next stage we're trying to investigate. That is, they're forming some kind of annulus, and there might be some correlation, some dependency. Once you have one activated, it may impact the probability that the other one being activated. So there is new development in this direction. Okay, but so I wanted to show this because in this program over the last 4 or 5 months, we've seen a lot in terms of simulations, but in terms of analysis of time and getting asymptotic expression, the methods are really different from just the Gillespie algorithm. You have really to attend to the analysis of Markov chains. And what was important in all of these cases is to put the right boundary conditions, and then the rest is not very hard. It's routine algebra. All right, so let's now move on. I would like to discuss, so today we are moving into more and more into biology. I would like to discuss now telomere. So I will explain what are telomeres. I would like to discuss a model about telomere change across cell division. And again, it falls into this framework of Markov, not Markov process, we're going to have Markov jumps. So let me state the question and the biology. So telomere dynamics across cell division. So this we started a couple of years ago, the following question. So you know that when a cell is born, it can divide a certain number of times. So this is cell, let's say time 0. I call time 1, the different time I increase the time when the cell divides. So it is known that after a certain number of divisions, the cell cannot divide anymore. This is called senescence. And that's it. The cell do not divide. As you know, in cancer cell, this threshold, this event can sometimes it do not happen correctly, and the cell continue dividing. And this is why cancer cells start to invade and create all the damages. So the process by which a cell stops dividing is not fully understood. It's under real investigation, especially what are the mechanisms that are involved to tell a cell not to divide. For example, for yeast, you can have a certain number of division, some 60 division, and after some time it stops. But there are variability. Sometimes you can see a cell that can have 90 divisions and everything is fine. So what defines the threshold by which you stop dividing? So let me remind you that 2012, the Nobel Prize in Medicine was given for answering this, partially this question. And the key answer to answer this question was the length of the ends of the chromosomes, which are called the telomeres. So you have basically in the nucleus, you have the chromosomes that are, the organization of which is still unclear, but basically they are anchored here. This is called at the centromere. And here the ends here are called the telomeres. So I would like in the class, maybe class the next time or the time in 2 weeks from now, discuss that actually in yeast, these telomeres, you have 32 of them. And when people ask, where do you see them? Where are they? Well, the answer was they are located on the surface of the nucleus, and more important, they form clusters. Now, these become a very interesting questions in clustering that allowed us to revisit all the Swolochovsky equations for clustering, because what people found is that they could form a cluster of, you could see 4 to 5 clusters in each cells. And the question is, why do you have clusters? What is the mathematics of clustering?
So David, that's known, that telomeres are always fixed to the surface? Or?
No. This was it was for yeast,
Okay.
and it was for, let's say, some kind of normal conditions.
Right.
Now, people did experiments during quiescence, that is when the cell is under stress. What happened, and so, of course, you can, if they mutate a certain protein, which is called the SEER protein, it's called SEER3, if I'm correct, then these are not anymore attached to the surface. They come inside, and inside what people have found, and this is still under investigation, they can form a big cluster, a huge cluster. And again, those properties like coagulation, fragmentation, they come, they bind, and they can unbind. Of course, what you have to understand here is, what is the meaning of forming a cluster? If you have 2 telomeres that comes, they can, one is coming, a second one is coming. Now, how do you put the third one? Is the third one going to bind to the 2 of them? Or would you have something like this, that they come as the finger in your hands, they come nearby like this. So when you have a break, you just break uniformly like here. Or if you have 4, you can bind, you can dissociate this way or that way. Or so this you have to make assumptions. But I won't discuss this today. I wanted to discuss this. Then you can study here a model of coagulation fragmentation with 32 to ask the following question. Suppose you have Brownian particle on the surface of a sphere. You have 32 of them. They are moving by Brownian. If they come, they can collide. You wait. What do you see? What's the steady state? Is the steady state a single ensemble of telomeres? Or do you have severals? And if you have severals, what is the mean size of the cluster or the mean number of telomeres per cluster? And there was also an important question, is that if telomeres can exchange genetic information, then you would like to ask the following question. What is the time 2 telomeres are going to spend in the same cluster? It's like the pair correlation function. So if you have, let's call this is number 1, and this is number 3. How long are they going to stay together? While many events can happen, because you can have a telomere that was nearby, that went off. So the cluster may change, the dynamic of the cluster, its identity is changing, but you don't care, because what you care is really about having the 2 specific that you have in mind in a cluster. So this was a tricky question in the combinatorics. So I'll postpone that for later. All right, I'm explaining where the problem is coming from. So you have these telomeres, and now I'm not discussing the spatial component. I'm now going to just forget about the geometry and discuss about the temporal aspect of it. So what happened during a cell division? During a cell division, if you have the end of the telomere, the end of the chromosome, so this is the chromosome, and this part is called the telomere, so it's more fancy, it has some kind of shape. I'm not going to enter into the details of the structure. And the idea is that if you are at stage K, when you go at stage K plus 1, with a certain probability, this has been diminished. The number of base pair here has been shrinked. So this is what can happen usually. But there is a process in nature which does the opposite. So this is more or less, you can see this event as systematic. Each time you have a division, you shrink. But there is a rare event which involves a protein called the telomerase. And what this does is that it can increase the size significantly. So this is like the usual, but sometimes, rare events, you have a big one. And of course, this is, you can think now, if you have this will maintain the length, this prevents the length to shrink. Why are we talking about the length? Because, and this is why this Nobel Prize was given, it says that basically what people were looking for years is that the clock of a cell is maintained by the length of these structures, which means that you shrink, you shrink, you shrink. If you reach a threshold, as we discussed before, if you reach a threshold, that's it. You stop dividing. There is a process which is unclear, unknown. I mean, probably it has to do with all this molecular pathway inside the cells. That the cell knows that it has reached the threshold and it will not anymore continue dividing. There is no next generation. All right, so this was our starting point. So let's write a model of this to study, for example, there's one more thing that I have to add here. Which telomere are we talking about? Is there one specific telomere? So for example, in yeast, you have 32. So they all have their own life. That is, let's assume that during this process of division, you have the 32 of them, and what happens to them are independent from each other. So you can see them as independent processes, and either a telomere can have its length decreased, or sometimes, rare event, it is increased by this telomerase enzyme. So then came, and it's still not fully resolved, and this was the interaction with our biology partner, is that the cell should be sensitive to the shortest one. That is, you look at your 32, and you are interested in the shortest. Because if the shortest has basically started to eat its genetic information, that's it. The cell doesn't want to reproduce if the genetic information has been damaged. You don't want to do that. That's why during double-strand DNA break, and the cell has broken, I mean the DNA is broken, if the DNA is not repaired, the cell stops whatever it has to do. It just has to repair. And if it doesn't repair during a certain time, the cell triggers apoptosis. It's better for the entire body that this cell dies than it replicates with abnormalities. This is the biology. Biology is good to kill, to preserve the ensemble. All right, so now we started to look at this question by saying, all right, let's model this, and this is what I would like to present now. I'll introduce the model, and then during the second part, we'll make a break after this, we will look at the analysis. So what is the model? The model is to look at the length. So let's forget now that you have 32, because we said that they are independent. Let's look at what happened at the length of the telomere at division n+1. So think about a Markov process. You look at the division, the length just after division ln. And what can happen is that it can decrease. I have to have the right, it can decrease with a fixed length a after each division, with a probability 1 minus p that depends on ln, or it can increase, and now this is a random variable. It can increase, and the mean of this variable is much bigger than a. So the probability of this psi equals y, we took it as an exponential law, py. And so I'll give you the values. So the values here are given for yeast. For other organisms, they are probably different. So because we worked on yeast, we had to find out all of these values for yeast. So p, the p here, which is your exponential parameter, was equal to 0.026. And so the mean number of base pair that was added was about 40 base pair that is added. While A here in average, it was 3.5, between 3 and 4. A was of the order 3 to 4, so we approximated it at 3.5. With probability 1/2, it was those guys. So this is about 10 times. So this is really a much bigger growth than the decay. And now, what was interesting is that we could find from data this PL. So we had points. So the probability of one of these events to happen was just decaying. That is the probability, the PL, when you shrink, this probability is increasing. So when you reach this state, basically the cell wants to prevent you to go drastically to shrinking without getting a chance to elongate. So the further you come to death, basically, the more chance you have to survive. And actually there are diseases where the telomere is affected, and it leads to accelerated aging in people. So this is a very hot topic to understand, in biological and genetics, what are the genetics disease associated to telomeres and cancer. This is very hot. Of course, we're not going to cure cancer by integrating by parts, but the idea is to give some understanding about how you analyze the data, what's the model, what can you predict, what happens when some parameters are changing, because sometimes it can affect with drugs some parameters in the model. All right, so for P of L, we just fitted, this is fit, you take data, you just fit a curve, and we fitted 1 plus 1 plus beta, L minus L0 for L bigger than L0, just, elementary function, and 1 otherwise. And beta here was, again, I give you parameter because usually nobody cares about parameters, but I think it's in the parameters, especially when you see small and large, that you can do then the calculation, the approximations. And L0 was 90 base per. All right, so I think with this, I'll give you one more information about, with already this, you can calculate, for example, what is the mean length. That is, suppose you wait and you have steady state, you can calculate the mean L minus 1 minus Ln. You can average this. So now you know the probability for this to happen. We know the distribution for this. And basically it's minus L multiplied by 1 minus PL plus 1 divided by PL. And so from this, at steady state, when you're not growing and not diminishing, so Ln plus 1 equals to Ln, you can get an average length of 350 plus minus, I forgot what, I cannot read anymore what I wrote, I think 102. All right, so now what are we going to be interested in? Suppose, all right, we have the model. We have information from data. Basically, all of the parameters now are fixed. There is no unknown, no model to fit. We got everything from wherever we wanted. Now, let's wait. You start, let's suppose you start with an L0 and you wait. What's going to be the steady state distribution of the lengths? Where are they going to be, how are they going to be distributed?
Can I ask you a question?
Yeah.
That L equal 350, is that a stable point that if you start there, it sort of stays there? And is it a stable point? Is it
stable?
So,
That's right.
So it's stable to small perturbations.
That's right.
Okay.
That's right. But usually you start, let's say, you start with a very long
I
telomere,
understand.
and then
Yeah, I mean local stability.
Yeah. So when you arrive here, this is stable. When you're here, you don't move. I mean, yeah, it's stochastic. So here at this point, you know, you go around and then you come back. Basically, when you reach in this region here, you have some kind of, this is where you find your steady state. You have a balance between decreasing and increasing. Although you have to imagine your process, you decrease all the time and then you have a big jump. So there is this large fluctuations. But this is the mean field steady state.
And there's only one
Yes.
fixed point? Like
Yes. Okay, so now here are the questions that I'm going to write down the questions, and then we will do the analysis after the break. So here are the questions. So what is question number 1? What is a steady state distribution? Second question, what can we say about the distribution of the shortest? So what is the mean length? Mean length of the shortest. So shortest among the 32, right? Among the 32. And now, so the debate in the field was the following. Is it true that this is the shortest? Why not an ensemble of the first, of the second? So we thought about doing the following calculation. Not that it answers exactly this question, but we said, is it possible that there is a gap, a statistical gap? You know, it's like when you solve partial differential equation and you look at the eigenvalue of your operator. You can have what is called a gap between the first eigenvalue and the second one. And usually this gap is interesting because it captures some inherent properties of the system. So we said that if you have the shortest, so this is the length here. And so you look at the first, the shortest one, which is here. Is it conceivable that if you look at the mean and if you look also at the variance, you have a region here, an area, and this area is it's separated. That is, what does it mean separated? It means if you calculate the distance between the shortest is the mean length of the shortest, and the mean length of the second one. That is, this is number 1, and now this is number 2. That you have indeed a separation, that this guy doesn't fall into the fluctuation of that one, because if it does in the fluctuation of that one, then it could be more complicated, that the cell might not be waiting that the first one has reached a threshold, but probably to the first, second, third, an ensemble of them. So what I'm going to show with the statistical calculation at the second half is that indeed there is not a spectral but a statistical separation between the mean of the first minus the mean of the second. This clearly separated, and the variance is smaller. This is what I'm going to show. So there is some kind of separation, right? So we will discuss this, and there is a third question that I don't have time to answer. I will not answer in this class. Is what is the mean time to a threshold? Because now it becomes a mean first passage time problem, where you impose a threshold at some point in this jump. So you have a jump process, and you want to calculate, to estimate when are you going to go over this. Let's say you fix a number what you think is critical. How long do you have to wait, and how this time depends, for example, on B. So the parameter beta here account for the effect of the telomerase. So if you change beta, it's like changing the concentration here. So really all parameters can be connected to the biology. All right, so let's break here. And after the talk, I'll show you how you can solve this in the continuum. This is very well-known technique. It's called the Takacs equation. This is a very well-known approach that has been used in queuing theory for the last 30, 40 years. And I'll show you that you can have an explicit expression for the distribution. And we will calculate using Laplace methods the mean length of the shortest and the second one, and we will see the separation. All right, so let's break 10, 15 minutes. We'll have to come back, and I have to leave at 4:45 because I have to pick up my kids from school. All right. Polymer, which is bit connected by spring, this is alpha equals 1/2. Usually for diffusion, we have 1, here we have 1/2. And so here is the question. If you prescribe alpha, this is prescribed, this is 0.43120. Is there a polymer model, and how do you have to build a polymer model where you prescribe this exponent? This is given. Why this question is become important? Because we had to analyze all the tracks of single particle trajectory, and we could find there was a spectrum of exponent. Now the question is why? So to look at this question, we decided to reinvestigate Rouse polymer, theory of polymer, and see how do you have to connect a single monomer with long, not necessarily the neighbors, but with longer ones, so the idea is to have an algorithm, given alpha, tell us what are the connections. So that with this connection, so we're talking about long-range connections, not anymore neighbors, then you create this anomalous exponent.
So this is a sort of distribution on the length of the segments.
That's right. So now you have, this is, instead of having, let's say, a factor, let's say a certain number for this one, you modify the one here, and now you start to connect with the second neighbors and the third neighbors, and they're fixed. They are fixed. You fix them forever, and then when you look at the exponent of this guy, not the extreme because they behave a bit different because they don't have connections, but if you look at what happened inside, you can see that the alpha is the one you prescribe, and I will explain how you construct this.
And presumably you could have many constructs like that? Or?
Maybe. We know how to offer one.
Okay.
If you have others, probably there are many ways to do that.
So it's some optimization procedure.
I will show. I will go step by step.
Okay.
It's about calculating variance of the motion of this and see how it depends on the neighbors, and see actually the procedure is you diagonalize the matrix and you use the eigenvalues, and instead of having the eigenvalues of the Orstein-Uhlenbeck operator with a certain power, we change the power and see how what's the consequence of the connection. And this power is exactly, some function of this power is exactly what you get in this exponent. So we could then go back and change the power here and explain how this is reflected in this long-range connection. I would like to discuss this. I'll try to go to this today. And again, this class, the next class, and the class after, I would like to spend really 3 classes about the nucleus organization, telomeres, clustering, chromatin organization, how do we analyze single particle trajectories, conditioned on the fact that you know that where you're observing lives on the polymer. It's not just a particle that is Brownian particle, it's something that belongs to a chain. Now, if you know this a priori information, you should be able to use this information for your prediction about what kind of alpha you're expecting, what are the forces you are expecting. Another question that I would like to discuss is, suppose there is a force on this one. There's a force. you observe this one. You observe the trajectory of that one. Can you, from the trajectory of that one, reconstruct the force that was exerted here? So obviously you have to deconvolve between what happened between the monomers in between. So this is the second question that I would like to go into all of these type of questions. It's 2 to 3 more classes. All right, let's discuss now a question also of interest, which is the distribution of the length. All right, so for this, the idea is to see that there is a difference, that this is small, and this is in principle much bigger, 10 times bigger. And so now we're talking about part 2. So this was introduction. Now we are part 2, which is really analysis. So for this, we are just going to rescale things. We're going to call x equal L minus L0 divided by R by A. Lambda x is equal 1 divided by 1 plus beta x. B, so those are normalized variable. B is A multiplied by beta. And we're going to normalize also this exponential probability. So the probability of the scaled variable psi equals y is theta exponential minus theta y. And theta here is this is just A multiplied by P. So you divide by A, so you have to see each. Basically, your process is you divide everything here by A, and you see how this propagates everywhere. So now the idea, I want to inject all of this into a time-dependent process. I call it delta t, the time in between two divisions. So let's rewrite the process. Instead of calling ln plus 1, I'll call it xt plus delta t. T is the discretization, I suppose, of the time, and we're going to write it x of t minus delta t, and x of t here plus psi. Now this is probability 1, the rate, minus lambda x delta t, and this is with probability lambda x delta t. And in principle, delta t here is 1. Delta t is 1 if you differentiate, if you divide everything by A, you get ln minus 1. This is a new variable, and so we have changed it according to psi divided by A. Why we're doing that is to inject everything back into some kind of continuum process. So now, if you look, did I say what is lambda x? Yeah, lambda x is given by here. Okay. So now, what's the probability? Now we're going to write the equation for the transition probability. So let's see what happens between two divisions. So how do you go from time t to t plus delta t? Let's assume that you end up with a length x. So what happened? Either you started at a length x plus delta t, and with a probability lambda x delta t you increased. So we started with a probability of having x plus delta t and time t, and with this probability you made your transition. Or And then we have to sum because it's going to be a distribution. You were somewhere with x minus z, so you are, and then you have to add something of size z to arrive to x. So multiply by wz, multiply by the transition probability, which is this one, which is lambda. Here I have to add transition probability, which is lambda x minus z multiplied by delta t. And then you have to integrate over all the z from 0 to x. Right, because you can only add. Okay, so now if we have to do a Taylor expansion of everything in delta t. So this can be written as probability of x and t plus the derivative dp dx multiplied by delta t plus something that you can neglect in delta t. This I can always approximate as lambda x plus delta t. Delta t, I suppose, is small, and you have delta t multiplied by delta t is going to be squared. So this can be approximated only as lambda x. I can write it lambda prime x delta t multiplied by delta t, so I get delta t squared here. So this will be negligible compared to this one. And this guy can go in here, and I will divide everything by delta t. So what happens when you do all of this? So basically, the idea again, you have this systematic shift. It's like a drift. This is why you are going to get some kind of hyperbolic equation in one direction. So again, let me please double check that I'm not making any mistake in the calculation. So this is x t plus delta t minus p of x t, and now let's divide by delta t. So I have moved this guy in here, and I divide by delta t. So then if I divide everything by delta t, the delta t here goes away, and I'm left with delta x, delta p here, and this is multiplied by 1 minus. So this stays, so this is, what did I say, dp dx, x and t. All right, now next, a term, I have p, yeah, is there, so p multiplied, I think I have the term p multiplied by this, divided by delta t, that should stay. So we have minus lambda x, p x of t, plus, and then it's like a convolution, so I can invert if integration dz. If I'm integrating this dz, I can just write y equal x minus z, so I can either put this in here or leave it like this. What am I doing? Yeah, so we can always rewrite this. It's integration from 0 to x of lambda y p x p no. Yeah, py t multiplied by w x minus y. And what is, how we defined everything? Yes, the y here, I'm sorry, the y here corresponds to the probability to this here. So this is p, right? This is the probability of making the jump. So this should be py. Okay, so this, when delta t goes to 0, this is exactly the limit of this. When delta t goes to 0, this is exactly dp dt. So you get what is called partial integral differential equation. So dp dt equals, yeah, this is correct, lambda x. So this says that if we know how to solve this, basically we'll get information about this transition probability. So the time dependent, we didn't look, so because we were interested in the question number 1, which is a steady state distribution. And the steady state distribution is you take dp dt equals 0. You impose that your distribution is normalized to 1. Because obviously this is up to a constant. When you have this is 0, so everything is linear. So up to a constant, you will have your solution. And now, actually, I'll give you the change of variables. So this is a bit analytically not that interesting, but I'll give you the changes of variable. If you just do this, and we call P tilde the solution at steady state. This is a steady state solution. So let's call it F tilde. So F tilde of x. Basically, the new variable here, you take this as a new variable, right? And now we have to replace. So lambda xf, this is u, so it's going to give you minus u, plus integral from this is exactly uy. W, this is the exponential here, so there is some theta somewhere. Theta, and then you get exponential minus theta or plus theta y, and here you have the x that is exponential minus theta x. You have to integrate from 0 to x, and then differentiating dp dx, which is df dx. So f tilde of x, this is u of x multiplied by lambda, but lambda is 1 plus this, so 1 divided by lambda is 1 plus beta x. 1 plus beta x, so it's very easy when you differentiate this. This is just the derivative of this plus beta u, correct? So you just plug this into here. What u prime 1 plus beta x. And here we end up plus beta minus 1 u plus, I don't know why I don't have the theta. All right, so I'm not going to be equal 0. So now this is becoming just calculation. I will skip some steps. But basically, what do you want to do now? You have u integral of u, and here you have something with derivative. So you just have to put this term in here and just differentiate one more, and you get a second order differential equation in u. You get u second, u prime, and u. And this you can solve, and this is associated to what is called the degenerated hypergeometric function. Okay, I think it's not that interesting that I do the calculation. Just wanted to explain the method that you arrive to here. And you can solve this equation, and you'll see you end up here with a distribution, a non-distribution. All right, let me skip the detail. I mean, I can just differentiate and let me skip this. It's not that interesting in terms of just calculations. So when you do all of this, and you use that, of course, your solution has to be bounded in L1. Fx is actually what is called, this is called a gamma distribution, which is basically, so f of x actually here. Okay, let me write it down. Theta plus 1 over b divided by theta, correct. So this is theta x plus 1 over b to the power 1 over b exponential minus theta of x plus 1 over b divided by the gamma, incomplete gamma function, which is 1 divided by beta plus 1 theta divided by b, where the incomplete gamma function, so the gamma function, the incomplete one, is defined by gamma s and x with the integral from x to infinity of ts minus 1 exponential minus t dt. And b, we have, this is here, a multiplied by beta. So this answers the first question. What is the steady state distribution? Right? You can have everything. It's given in terms of the parameter of the problem. We have, so the gamma distribution basically, this is something like this, right? You can go, so here there is a shift. It doesn't start exactly at 0. At 0, this is something, so you have basically something and it decays exponentially. All right, so now let's go to the more interesting part in terms of the analysis. Let's discuss now the statistics of the shortest one. Question number 2, statistics of the shortest telomere. The statistics of the length of the shortest telomere.
But with this already curve, could you integrate up to L0 and then determine what fraction of cells are expected to die at steady state, in a way?
Well, the reason that, you mean ignoring the fact that you're interested in the shortest one?
Yeah, because this already gives you...
Yeah, this gives basically the distribution of the length, but...
Is this for one?
This is for one, because let's assume that they are all independent. So basically it gives you the distribution for all of them. So let me show you what we did with this. So we were indeed interested, you have 32, and now this is, so you have 32 in here, so you distribute now 32 of these guys. You shuffle everything and say, all right, where is the minimum one? So this is really the statistics we're interested in. This is called extreme statistics. There is, in statistical physics, there is a branch that is going into that direction to look at extreme events. So basically this is, we can call this extreme statistics, but this is really looking really at the minimum of the distribution. So we have, let's say, x to x32. Let's call x1, x32 the length of them. So let's call it y. Usually this is called like this, x1. Let's assume we have, I call it 2n. We have 32, I'll call 2n of this. Now, what is the probability distribution for y? Now, this is not very hard because we just calculated the distribution function for any of them. So take the probability distribution, take 1 out of the 32. So we have this 2n. Let's take the distribution, so we call it f of p, f of x, that the probability that the shortest one is equal to x. And necessarily, the 2n minus 1 have to have a length which is larger. So what is larger? You have to, if you integrate this from 0 to any x, from 0 to x, this is the probability that it is less than x. So you want 1 minus this, so you have 1 minus f of x, and the probability, because they are independent, that they are with power, it's power 32 minus 1, so let's call it 2n minus 1, and f of x is 1 minus, so this probability that is older, shorter, less than x, which is 0 of x, f of, let's call it f of y dy. Okay, so now we're in business. This is elementary analysis now. How do you calculate the length of the shortest? We have the distribution for the shortest explicitly, because we spend a lot of time calculating the distribution for any of them. So what is the mean? What is the mean of the length? So we have to look at the first moment, right? So let's call it y, mean like this. So this is just y, d dy of the probability that, let's say, the mean length y is less than y. This is by definition, this is the moment. This is the first moment, the length, according to this distribution. So this is the distribution of what is written exactly here. This is exactly fy of x. But now you can, if we integrate by parts, this, you'll get exactly, so you integrate by parts, you differentiate this and you integrate this. And what you get, you have to integrate these guys. And this guy is going to be exactly sum from 1 to infinity of 1 minus capital F of y to the power 2n dy, which is f of y is here, the integral of f, which is what we calculated. So now this is interesting because let's assume n is large. Then how you estimate this integral? You just do Laplace methods. Laplace method in dimension 1, so this is very well known. And from this you can have an estimate, and the estimate is gamma 1 divided by alpha alpha gamma of alpha to the power 2n, 1 divided by alpha, where alpha is 1 plus 1 over b. Alright, you don't have to memorize this, but now we plug numbers and we find out that n is 184 plus minus 25 Leicesters. So this is the mean, then we calculate it with y squared to get the standard deviation. We did Laplace methods when it is squared. There is an additional term here that you have to be careful. And this is what you can get. Now, next stage was to do the same for the mean first passage, for the length of the second shortest. So what is the length of the second shortest telomere? How do you calculate this? You need to be above the first one. So this, basically, you have your distribution of the lengths. You have the first one, which is here. So the second one, the probability distribution has to be, let's say, equals to an x. So let's call, what do you call f of z? This is for the second. So this is now the second shortest telomere. So f of the probability to be at x, you choose one among the two n, and then you have to choose the one that is going to be shorter. So shorter means that the length has to be distribution is f of x, and then the rest, that is the 2n minus 2, they have to be in this region. So this is exactly this to some power 1 minus f of x to the power 2n minus 2. 2n minus 2, is that correct? Yes, it is. All right, same methods, you calculate, you can now use, you take z, you take the mean, you take the interval from 0 to infinity of your x, fz of x, you integrate by parts. So you do the same type of methods. And what we found is that z divided by l, d divided by y, the mean, there was something universal, which was 1 plus 1 divided by alpha, where alpha was this 1 plus b. And so what we found is that the mean of z, when you plug all numbers, is 207. Now this was great. Why? Because you have now the minimum, what we call it y. This is the minimum one, the minimum size is here. Now you have your variance, plus minus 25. So how much is 25 plus this? This is about 200. And here lies our guy, z. Suggesting, again, this is just a suggestion, that probably the size of the minimum one, there is some kind of statistical separation which can be linked to the parameter of the problem. That this is, I would not say far away, but it is not, at least in the variant. This was important for us to check. That probably the cell feel or sense the shortest one when it reaches some kind of threshold. And this is the shortest that may be the key to trigger apoptosis. And not the couple of them, because obviously when this one reach a threshold, the other haven't reached anything, because obviously when you jump, you know, you do this -3 or this plus something. So if you are above this critical threshold, obviously the other might not be. So this was the reason we did this calculation.
David, are your values of L correct? Because L is up to 209, and 209 is obviously within the range of 207. I think your argument stands,
we are
just inside.
Okay. That's right.
Your argument stands, but it's just not quite right to say it's not in the range.
No. It's just in, you know, few base pair. Again, this is a suggestion. I'm not claiming anything. I'm saying from the statistics, there is no like a gap separation that you can have in eigenvalues where you have one very close to 0 and there's a gap. Here the gap is statistical. That is, you are not in the variance. This is the idea. You are not in the variance. There is no clear theory on this, so we had to make up something. That's all what we claimed. But what was interesting is to come out with numbers and to link this with to parameters, and to show that the statistical analysis could say something about the extreme statistic about the minimum, the shortest one. The next paper will be about when. That is, you ask the question. Now if we go to mean field, in mean field you have a potential. Now, where is the threshold compared to the minimum of the potential? Where are you going to put it? You have the choice. I mean, we don't know where it is. So we looked at the situation where you have...
What potential again?
I'm sorry?
What potential? I missed that. Well, what
you were asking, what you had in mind, I suppose, is that...
Oh, you mean like it has a fixed point issue?
That's right.
Okay.
When it comes to the analysis of this system, what you can do is you can derive, as you do in stochastic chemical reaction, you can calculate what is called the mean field. You look at the drift component for the stochastic equation, and you can see that the drift has the shape it's the derivative of a potential well. This first moment. And so when you're here, basically, it's like you are going around also with fluctuations. Now you are looking at mean field. And so still you are interested in when are you going to reach, so for example, suppose you put your threshold here, when are you going to pass this threshold? So what we found, we haven't published all of this yet, so that you start from a long telomere, you're going to fall into this, obviously, because you're
That's
falling
very quickly, right?
very quickly, although compared to what? All right, no, this is a good question. This is a good question. So you are going to go in here with a certain time. What we find out is that the time it takes to go from here to here is of the same order as the time it takes to escape. Now this is very interesting because then we calculated, again, we haven't published this, but the idea of all of this, it says that telomerase is saving you about 30-40%, or extend the life of the cell of about 30-40%. It extends the life of a cell of 30-40%. The time that you would, if you would have come to here, you're dead. Here you get stable, but the time to go from here to here is of the same order as to come to here, although this is much shorter. So you are basically maintaining in life close to the point where you are going to die.
So at the edge of stability.
That's right. Yeah, of stability to die. I mean, not die means senescence. Okay, I haven't shown this. This is much harder because this has to do with now calculating mean first passage time to the threshold for a jump process. And this is involved. It involved calculating Wiener-Hopf integrals, and this was not ready. Okay, so we have 10 minutes to start entering into 2 or 3 classes of the nucleus organization. And so let's not discuss. So the nucleus organization is a big story. It involves now quite a bit of experimental group from genetics, microscopy, to really characterize how the chromatin inside the nucleus is organized, and the normal and pathological condition under perturbations. So modeling chromatin organization. So for this, I would like to start with questions about polymer physics. What seems surprising in this entire field is that the simplest of all the polymers gave the best, is one of the best models for interpretation of data. So part 1, which is really, so the introduction is really about how the nucleus is organized, So you have your cell, inside you have the nucleus, and again you have chromosomes. So in mammalian cells, people have described territories. That is, if you have ever done simulations with a polymer, and the polymer repel each other, that is, or if you cook spaghettis at the beginning, or it depends what kind of spaghettis, but basically, you can see that they stay. This is what they see under the microscope and the interpretation is that you have territories. The chromosomes do not mix for some reason that are unclear. So the question was turned to the physicist: is it true that if you simulate the polymer with exclusion forces, I mean like Lennard-Jones forces, that naturally gets segregated this way? Of course, it's not true in yeast. In yeast, there is no segregation. In yeast, what you have, if you look at the nucleus, so this is the nucleus. Now this is the nucleus of the yeast. I'm not going to draw the cytoplasm. And you don't have this. You have, this is the centromere, as we have drawn before, and you have the telomeres. And the telomere form clusters attached to the boundary. Under some conditions, the telomere can even detach and form in quiescence like a big cluster. So there is a big debate about how the nucleus is organized. And so I have to tell you about one more type of experiment. So there are two types of experiments. This is the description of where the data will come from for this interpretation. What can you do to see what happened inside the nucleus? Well, using microscopy, people have tagged chromosome and they look under the microscope, and what you see, and you can collect trajectories. Now the question is, what can you extract from the trajectories? What can you extract? What can you extract from trajectories? So this is a big debate. And I mean, debate means you can extract the fusion coefficient, but we're going to see we can extract much more. You can extract some forces. You can extract information about the chromatin, about the polymer itself. You can extract information about the alpha, the anomalous exponent, let's say, before you do anything, and after you induce a break. So suppose now you do the same experiment, now you have a break here. Can you see the effect of the break in the trajectories? The answer is yes. Now we have a paper. We are trying to get this paper published. We're doing a mix of experiments. Not that we did experiments, but we analyze the data to try to understand how do you differentiate trajectories, what kind of statistical information you get before and after a break.
Sorry, break, you mean like you stop the measurement?
Break means you do a break. You cut double-strand DNA break. So you have your DNA. And then you cut it like this. It's really a break.
Okay, sorry.
Single-strand DNA break is much easier to repair, but double-stranded DNA break, this is much harder. There are two pathways. I don't want to describe them now. Basically, either you glue them, or if you have missed some information, you have to find a template to get back the information. And this is much harder because then you are involved, the DNA or the chromatin is involved in a search process. So again, you don't search with a Brownian particle like we did in chemical reaction to search for a target. You search like the head of a snake is searching, and it drags its entire body toward the target. Okay, so this is one type of data, which are called, so the first type of data I've described, this is called single particle trajectories data, which give dynamical information. So people perturbed, you can do genetic information, knockout, you perturb some genes, you perturbed, and then you go back to the trajectories, try to extract features, and then you compare features before and after. So this is the first type of data. This is called single particle trajectories. This is what we just described now. The second type of data that is going to be very interesting, and that has been the result of quite a lot of effort, is to have a frozen picture. So you have your DNA, you freeze it, and then you look at the following question. Can you obtain, fix, let's say monomer, let's call it I, and fix another guy, J. Average, so get information about the distance between I and J, the encounter probability, and do that over all I and all J. Basically, you end up with a distance or a frequency matrix of, so you get your, let's say, let me write it as a distance here because this is clear. Now you get a matrix, and if you discretize this with a certain scale that you fix, you can end up, you can get in the end a matrix of 1 million by 1 million. And you do that, you average over this over many cell realization. So what you end up with, what is called the frequency of encounter. That is, you look at the fraction in the ensemble of cells that I was close to J in, let's say, 1 million cells, and you look at this frequency, and you do that for I and J. Now, the big question in the field, what can you reconstruct from this matrix? So what can be reconstructed? What can be reconstructed from the encounter probability matrix? This is unsolved. Probably there are many solutions. Probably it's completely nonsense because you average over many cell realization, and if the cells are in different configurations, you're going to have some kind of ensemble. What can you get from this ensemble? Well, it turned out that people have discovered many things with this. They discovered that they are part of the DNA or part of the chromatin, that they are forming subregion, and this is stable. So then you can try to reconstruct this subregion, why this subregion is stable, why it is together, and get more information about how the nucleus is organized. Again, for example, in yeast, if a gene is expressed, it can be on the surface. If it's repressed, it can be inside. So there is also a relationship between geometrical organization and gene expression. So now the first approach to let's spend 5 minutes on this, and so this is really part 2, which is what kind of polymer model are we going to use to describe, to analyze, to calculate. So for example, the type of question you would like to be interested in is, for example, as a function of distance here. If you let everything evolve according to Brownian motion, how long do you have to wait to form a loop? Right? So let's ask some questions. So the questions that were of interest that I would like to discuss, and for this, we need a polymer, we need a model for that. So the question is, what is the mean time to form a loop? Form a loop means by Brownian motion. Second, we are going to discuss is when you have the mean square displacement, which is the average, let's say, of xt plus delta t minus xt, and your average squared over your realization, it goes to delta t to some power alpha. What is the meaning of alpha?
David, can I ask, what do you mean by a loop? Do you mean with your diagram where I and J come together?
Yeah. So a loop. So suppose you have a polymer. Suppose let's just discuss about the two ends, A and B. How long it would take for B to come in the ball of, let's say, radius epsilon, and epsilon you can vary, but this is something that it's supposed it is fixed. How long it takes for this guy to enter into this loop for the first time? Right? And can you write an expression? Can you find it analytically?
Okay, is that how you construct it, or are you thinking of two epsilon regions around A and B and then coming together?
That's more or less the same. If you can solve this one, you are happy. And it, of course, I put the end, but it can also be inside, as you can have in here. How long it takes for this guy to come in this close proximity for the first time, and then it separates again. So to have this will give us the rate of the information about the encounter probability, the encounter frequency matrix. But now we want to have this using a model, using a polymer model. So we know how to solve this for Rouse polymer. What is beautiful is that nature seems to be using, I mean, the Rouse polymer seems to fit quite well, although it has some little discrepancy, but it's very good in terms of analyzing the chromatin at the scale that we have under the microscope. So what is the meaning of alpha? And then there is also how do you construct basically estimators for parameter you want to extract, for example, how to extract from information, how to extract the diffusion coefficient, of course alpha. You can extract also, and this we will see, forces, and I'll explain how to do this. That will characterize the motion of the chromatin or the motion of a single locus. And so for this, to answer this question, so this will take us at least, I think, 2 classes. We need to introduce a model, which is the Rouse model. It's
So could I just ask a minute? So, I mean, we know that the DNA can wrap around histone complexes, and then these things can separate, and this is one of the mechanisms in epigenetics, right? So, have you thought about these things?
well, nucleosomes, this is, the scale of this is really the scale where nucleosomes are not distinguished.
Yeah, no, I can see.
Because then if you want to add nucleosome on a polymer model, this is very interesting, this is very nice actually. You have first to have, let's introduce the polymer model. So here we'll discuss mostly Rouse, although I'll explain other type of polymer. So you have beads that are connected by spring. So you start with bead number 1 and 2 and 3. So this is really of course graining of what? What are we talking about here? As you said, you know, David, you're talking nonsense. You have nucleosomes where the polymer, the DNA wrapped around, and then there's another one, and then the other one, and then it goes like this, and this is what you can see in textbook. It goes twice, and then twice, and then twice. Here what we're talking about is something abstract. This can represent any number of base pairs. So we have to define what is the scale at which we are going to use this. For example, we can say, I'm going to collapse 3 kilo base pair in each bead. So then I don't see this picture. If you want to see this picture, this is very hard. Then if you want to make any simulations, people are doing this. It's very hard work to look at simulation. Then it's basically a molecular simulation. You try to look at the interaction, and then you don't go very far with the time scale of your simulation. If you want to have long range processes such as looping is a rare event. You know, it's the time it takes for 2 to meet again. So this is a rare event. No chance that if you add or if you start with this complexity, you answer this question. This is for example, we looked, this is also in published work, when you send UV light, you create damages. The damages turn out to lead to translocation of these nucleosomes. That they are pushed away, or maybe they're falling. Now you need a different model. You need to add on this a model for the nucleosomes. Okay, if I have time, I'll discuss a bit this. But so now the idea is how you write now equations for this Rouse polymer. So let's start with this. So this is very classical. In the Smolachowski limit of the Langevin equation, so x dot equals square root of 2d. This is a classical stochastic equation for one single particle that is moving with diffusion coefficient d. Now it's the Rouse polymer is very easy to understand. You have bead xk. It is connected to bead xk minus 1, bead xk plus 1 with a spring constant. And then you write down your stochastic equation for this guy, which is x in the Smolachowski limit of the medium. This is, let me write it, 2rk minus rk minus 1 minus rk plus 1 plus square root of 2d. Let's suppose this is the same for all of them. And this is the white noise. And you have to be careful at the end because this guy has no guy on the left and this guy has no guy on the right. Now what do you do with this? Now it's becoming more complicated than the simple stochastic equation for one particle. It turns out that you can do a lot. And so let me explain in a few minutes here how from this you can start now construct polymer with a given anomalous exponent. So I have to tell you that if you look at the anomalous exponent of a monomer, okay, of t squared, and you look, let's suppose at time 0 this is in 0, this goes like it's t to power 1/2. Diffusion, this is 1, here this is 1/2. So this is the t small, it behaves this way. So now let's see how, a method of construction of a general polymer. The idea is to start by using a diagonalization of this matrix here. So you can always write this is a matrix. You want to diagonalize this matrix, look at the eigenvalues, and look at this equation in the space, in the eigenspace. So this is a three-diagonal matrix, and this is well known, this has been worked out 40, 50 years ago, that you can use the following transformation. You write another set of coordinates, UP, is Rn alpha P of n from n going from 1 to capital N, and alpha Pn are given, so this is really an orthogonal matrix. This is 1 over square root n for p equals 0, 2 divided by n, cosine n minus 1/2 pi p divided by n. All right, so this is, you change coordinates. This is the Rn, you multiply by this matrix, and now you want to see what is the stochastic equation satisfied by this UP, so you just replace here. So what do you win by doing this? This is a way to diagonalize the matrix, and you end up with an ensemble of an equation which is dup dt, just differentiate this with respect to time, equals minus d kappa p up plus square root of 2d, and then you have a new noise, whatever, okay, let's call it wp dt, where kappa p equals 4 kappa sine pi p divided by 2n. This is important, squared. So this is true for p going from 1 to n minus 1. For 0, u0 is really only the center of mass, which I don't care, because basically, if you have a polymer, the polymer will move. So you can take the average of the motion, and this will give you the center of mass. Now p in the polymer, now you cannot have this translation motion, and this is basically what you have here from 1 to n minus 1. Right before we had something beats from 1 to n, so you can ask where is the missing one. So the missing one is just the Brownian motion of the center of mass, which I'm not writing. So I want to make sure that, yeah. So let me write down. So before I have forgotten, here there was a kappa, which is the spring constant. This is the kappa that is in here.
Don't you get additional vector in the noise term?
Yes.
Because you,
yes.
It's
But
an eigenvector of.
that's right. But this is equivalent to another Brownian noise.
I see.
But you don't care.
Okay.
That's why I wrote it tilde. It's a sum,
Okay.
and it's a Brownian motion, so it stays Brownian motion. What is important is this. And now in the 2 minutes that I have, I just want to explain how we move all of this. So what have we done here is nothing really. This is dUdp. So now I have to write one more thing, which is the so associated to this, there is one important quantity, which is the energy. So the energy of R1, Rn. This is simply the sum of kappa divided by 2, Rn minus 1, squared from 1 to n. And you can always rewrite this using the gradient of u in here, gradient with respect to Rk. Now here is the idea. This is in the Fourier space. I mean, an ensemble of Ornstein-Uhlenbeck equation. This is linear. Kappa p is given. So suppose now instead of 2, we changed it to beta. Beta different than 2. All right, you have the 2 here was crucial. Now let's put another exponent, and now let's do the inverse of this and see what kind of polymer you get. So this is the first part that we will do next week. Want to see what you will get here, and in particular when you do this beta here.
And the reason you're doing that is because that's the fractional eigenvalue of that Laplacian operator. I mean, this is a finite difference.
That's right. That's right.
But it's a Laplacian.
That's right. It's a Laplacian.
And so you take the fractional Laplacian of that, and you get that eigenvalue.
That's right. And now you'll see how this will propagate, and yeah, this will give us the fractional and how it will spread into the energy. And this energy. So if we, I'll take just a minute to write this because as Kevin started to mention, so in when you write this into this U mode, you get 1/2 of the sum of kappa p dp squared. This kappa p is equal to this. Now we're going to change it. So this, let me put it in square. Now we are going to change it to beta. Up can be, you can use now this guy to plug into here. And this is the energy. So when we are going to express everything back into the Rn, this, what is the interpretation of it? This is to explain that the connection is between the two neighbors. But now, when we will expand this, we will get exactly the new matrix, because with the beta, which was 2, you had all this cancellation. Now you don't have any cancellations. Now all of them are going to be coupled. As a matter of fact, you know, you can write it kappa p beta. So up is, let's replace, this is the double sum of r, let's call it nm alpha pn, rm alpha m p. So you have to sum over p over n over m. And now you are not going to get necessarily this, but you're going to get the coefficient in front of Rn and Rm, right? Because this is not going to be anymore, so the coefficients that you're going to get.
So it's not distance dependent anymore?
That's right.
Any two bit?
That's right. Because before, if you look at the coefficient A alpha m, this is sine beta pi p divided by 2n. And then you have to multiply by cosine m minus 1/2 pi p divided by n, multiply by cosine m minus l minus 1/2 pi p n. This is the coefficient that we are going to discuss. Yeah, and you have to sum over p. If you have here squared, everything will cancel. If you don't have, if you have a general beta, it doesn't cancel. So this is a procedure to construct this new interaction. So next week, let's stop here. Next week I'll show this, and then I'll show how you calculate now with this the new anomalous exponent and how this is associated to the beta. So to see the connections.
A quick question. Do you find general decay with the separation ij in the structure? That as the separation between I and j increases, the
Yeah,
the
there
coefficient becomes
is.
smaller?
That's right. That's the answer to your question.
Exactly.
It decays like very quickly.
Okay.
Yeah, you see. Yeah.
So it has a local sort of.
It's local. Yeah, it's local, but you need
Okay.
this stuff far
So
away.
it's like a local many body sort of thing.
Like in many. Sorry.
It's like a local many body effect in a way.
Well, you need what happened far away, otherwise you cannot construct. But it's true that it is small.
Yeah.
Is there,
It's a local, non-local model.
That's right.
but is there a characteristic land scale too, or is it like a parallel? So you
It's-
know,
Okay. It depends what we're talking about. You will see the t to some power alpha. Now, what is interesting here, the correlation function is still exponential.
I see. Cool.
That's the idea. Okay. Thanks.